Now it's time to talk about water chemistry. In this chapter, we're going to talk about understanding water chemistry and why it's important, and then we're going to talk about temperature, dissolved oxygen, pH, dissolved solids or conductivity, nutrients, and transparency. Those are all parameters that you could potentially monitor with our program. And then we're going to go over data sheet and methods of how to collect all of these different parameters. Alright, let's dive into understanding water chemistry. Water is something that's required for all life on Earth. It allows necessary biological reactions for life to occur, and it's something that helps carry nutrients and minerals to aquatic life, and it helps us carry waste away. Water chemistry is important to human health as well as aquatic health. It also affects the abundance and diversity of the aquatic life that lives in the water. And it's also important to note that changes in one parameter can affect other parameters, like an increase in temperature could also affect a decrease in dissolved oxygen, and so forth. We care about water chemistry because it's related to toxicity. Toxicity is the measurement of how poisonous or harmful a substance is to plants and animals. There's two different kinds of toxicity. There's acute toxicity, which is whenever an organism is exposed for a short amount of time to a particular substance, generally 2 to 4 days, and being exposed to this substance can cause lethal or very serious harm to the organism. And then there's chronic toxicity. Chronic toxicity is whenever an organism is exposed over a longer period of time, generally over their lifespan or at least a tenth of their lifespan. And it's harmful, but usually it doesn't cause death, but it can affect things like growth or reproduction. Another reason to monitor water quality parameters is to understand how the water relates to the standards that we set for it based on its uses. If these standards aren't met, then some or many aspects of what we want to use the stream for might be suffering. For example, currently the Niangua darter is an endangered species in Missouri. They need very specific physical and chemical criteria to be met in order to survive and reproduce that currently aren't being met in much of their native habitat right now. In order to get a baseline of understanding and data for areas that you want to monitor, we recommend that you take water chemistry 4 times a year, once per every season. And because flow and water chemistry are very interconnected, we also recommend that you try and get flow data along with your water chemistry data. Alright, so let's jump into our first water chemistry parameter, which is temperature. So let's talk about the effects of water temperature. So as water temperature increases, the amount of dissolved gas that's available in the water is going to decrease, specifically dissolved oxygen. The rate of plant growth increases with warmer temperatures, and the more plant growth means more dissolved oxygen because photosynthesis. The rate of photosynthesis from plant growth changes with temperature as well. And photosynthesis is important to aquatic ecosystems since plants produce oxygen only during the daylight hours. Certain pollutants can become more toxic at increased temperatures as well as modify the state of the chemical. And then in terms of metabolic rates of organisms, as the water warms up, metabolic rates speed up and organisms need more oxygen, including speeding up of the rate of decomposition. Changing the temperature range of a stream can change the whole ecosystem. Many organisms require a specific temperature range so that if the range is shifted to a higher one or a lower one, many species of invertebrates and fish will no longer be present and living there. Our water quality standards for temperature are based on organisms that can stand these different ranges of temperatures. Our warm water fisheries is our general aquatic life water quality standard, and that's about 32 degrees Celsius or 90 degrees Fahrenheit. Cool water fisheries are a little bit cooler, and they support aquatic life like smallmouth bass and rock bass. And then cold water fisheries are even cooler, so about 68 degrees Fahrenheit, and they're meant to support organisms like trout and hellbenders. So some ways that humans can impact or change water temperature is firstly riparian cover removal. So if a stream has a lot of trees along the banks and it's getting a lot of shade coverage and then that coverage is removed, it's going to allow for the water to be more exposed to the sun and therefore it's going to heat up more. Another way is by erosion. The more turbidity or suspended solids that are in the stream channel, the more the water can retain that heat from the sunlight. Another way that humans can impact water temperature is by industrial thermal pollution. So this is any industrial or power plant that discharges water into a stream that's warmer than the stream itself. And also you have impervious thermal pollution. This is whenever water is entering a stream after it's hit an impervious surface that's pretty warm and it increases the temperature of the water before it runs into a stream. Also to note here that many storm drains are connected directly to streams in urban areas. So this can have a big impact to aquatic life in urban areas if they can't withstand the increased temperatures of the streams. The next parameter that we're going to go over is dissolved oxygen. So it's really good to monitor for dissolved oxygen because it's critical for all life in a stream. Just like we breathe oxygen from the air, which consists of 21% oxygen, in the water it's about 5 to 15 parts per million, or 0.0005% dissolved oxygen. So there's a big difference, and it's really important for the aquatic organisms that live in the stream. Also to note that parts per million is the same as milligrams per liter. Whenever you measure for dissolved oxygen, you're going to be getting them in milligrams per liter, but it's the same. So how does dissolved oxygen make its way into the water? Well, this happens a few different ways. The first being just a tumbling action from waves going over rocks or other things in the stream. Kind of like the picture that you see here, the white water or the rapids, so to speak, that are going over the stream is what's causing that tumbling action to sort of diffuse the water into, or diffuse the oxygen into the stream, which is another way, diffusion from the atmosphere. As water in the atmosphere are close to each other, oxygen from the atmosphere will actually enter into the water itself. And then photosynthesis is a big one. So algae and other plants that undergo photosynthesis in the stream output oxygen as a byproduct of photosynthesis, and this creates extra oxygen in the stream for aquatic organisms to use. So specifically, why is dissolved oxygen important? Aquatic organisms need a certain amount of dissolved oxygen in the stream in order to survive, and whenever that is depleted, it can actually cause the death or an entire population shift in the amount of organisms that are present in the stream. This is due to the sensitivity that organisms have to the amount of dissolved oxygen that's there. So the water quality standard for dissolved oxygen is no less than 5 milligrams per liter, or that 5 parts per million. So we've kind of already touched on this a little bit, but there's some pretty natural things that can influence dissolved oxygen in a stream. Temperature being one. As temperature increases, the amount of dissolved oxygen that can be withheld in the stream decreases. And then flow, if the water level is really low and it's not flowing very fast or flowing over things, that can cause that tumbling, that turbulence that's going to incorporate the dissolved oxygen from the air into the stream. That's going to affect the amount of dissolved oxygen available. And also suspended solids, like we talked about earlier with thermal pollution. If there's a greater amount of suspended solids in the stream, it's going to create a higher level of ability for the water to be a higher temperature, because the suspended solids can hold more energy and more heat in the stream column. And then aquatic plants that produce photosynthesis, again, that's going to directly impact the amount of dissolved oxygen that is available in the stream. Humans can also influence the amount of dissolved oxygen that's in the stream. Firstly, the removal of the riparian corridor. That's going to affect the water temperature as more sunlight is available on the stream. So that's going to increase the temperature, which in that case would decrease the amount of dissolved oxygen that's able to be withstood within the stream. Dams are an interesting one. So water that's discharged from dams can be supersaturated with dissolved oxygen or nearly devoid of it. So it depends on where the water comes from in the lake. If it's taken from the bottom of the lake, it might be almost devoid of dissolved oxygen. But on the other hand, if the water comes from the top layer of the lake, this might be high in dissolved oxygen. In this case, the addition of more oxygen as it tumbles over the spillway can cause the water to become supersaturated. Too much dissolved oxygen can actually kill fish by causing the same symptom as the bends in divers when they ascend too quickly. If a dam is discharging into a water of the state, they have to meet water quality standards of 5 milligrams per liter at least whenever they discharge. There's also organic waste. Organic waste can affect the amount of dissolved oxygen as it breaks down inside the water. Bacteria use dissolved oxygen to break down the organic waste. In that case, it could deplete the amount of dissolved oxygen in the stream. Sources of organic waste can be stormwater urban runoff, septic systems, wastewater treatment plants, animal feedlots, and discharges from food processing plants. DIEL means daily. So this is the daily fluctuation of dissolved oxygen that you can see throughout the day. Here we have two different lines represented. The black solid line is represented with normal DO fluctuations that will happen throughout the day in a stream. The dashed line is called a eutrophied state of dissolved oxygen within the stream. So this is a stream that has had excess nutrients in it so that it causes a higher swing in dissolved oxygen. Why is the dissolved oxygen lower at 6 a.m. here than at 6 p.m. here? Well, it's lower because algae and other aquatic plants switch from photosynthesis to respiration at night and are therefore using oxygen, not producing any oxygen at all over the night period. So the only source of the dissolved oxygen during this period is a result of physical aeration in the stream as water tumbles over rocks and other obstructions. Now, if a stream is slower moving or very low in flow or pooled, that means that even less aeration is getting into the stream over the night period whenever algae and plants are respiring and not producing oxygen with photosynthesis. Let's consider also when it's summertime and the stream water may get really warm. Warm water can't hold as much dissolved oxygen as cold water, and simultaneously, warm water results in the metabolic rates of organisms being increased. The higher the metabolic rate, the greater the demand for oxygen in order for organisms to function. This also applies to bacteria who are constantly, both day and night, decomposing the dead plant and animal matter that falls to the bottom of the stream. The constant decomposition process requires oxygen. All of these factors together result in more oxygen being used at night that is physically added to the stream via aeration during the night. Therefore, it's normal for the amount of dissolved oxygen in the water to decrease as the night goes on, only to increase once again as the sun comes up and the plants start undergoing photosynthesis. However, if these processes occur to extreme levels, subsequent extreme fluctuations in dissolved oxygen, represented by this dashed line on the graph, can occur. Many factors could contribute to this, including removal of the trees from the riparian zone that would normally provide shade for the water and help control erosion. Also, if streams receive excess nutrients, like nitrogen and phosphorus from fertilizers running off the watershed, it may result in excessive production of algae. Excessive production of algae is often called a bloom and can cause extreme DIEL fluctuations in dissolved oxygen. If the previously mentioned factors result in a large dip in dissolved oxygen at night, you might have problems first thing in the morning. You can see on the graph where the dissolved oxygen levels at 6 a.m. are below 5 milligrams per liter and within lethal limits for aquatic life. In other words, organisms that require oxygen could begin to die if the dissolved oxygen drops below 5 milligrams per liter in a stream. This is especially true if the night is followed by a cloudy day. This is why water quality professionals try to take dissolved oxygen readings early in the morning so that they can potentially see this increased swing in dissolved oxygen throughout the day. Another helpful way that we can represent dissolved oxygen is by percent saturation. So dissolved oxygen concentration tells us how much oxygen is in the water. But how do we know if that's enough dissolved oxygen? So DO percent saturation tells us a little bit more about that. It's the ratio of the amount of oxygen present in the water to the maximum the water could hold at that temperature in the absence of any other influences. It's a little bit more meaningful than just the DO reading alone. So here's a graphic. For warmer water, less dissolved oxygen is able to be withheld in the water column because there's more energy, those water molecules are moving, so they can the oxygen molecules can make its way out of the water column. Whereas with cold water, more oxygen can be withheld in the water column because it's slower moving water molecules that help those O2 molecules stay within the water column. So, as water temperature increases, the amount of oxygen it takes to saturate the water decreases. Because of this, cold water can hold more oxygen than warm water. Here's an example. So if you get a DO reading of 8 milligrams per liter and it's summer and the water temperatures are hot, this could be a great DO reading. But in the wintertime, whenever the water temperatures are really low, a DO reading of 8 could actually indicate some problems for the stream. So DO percent saturation helps take the guesswork out of interpreting dissolved oxygen measurements, because it takes that temperature into account. Here's an example with a sheet that we'll provide for you and that's available online of the relationship between dissolved oxygen and percent saturation. If you obtain a dissolved oxygen reading of around 8 milligrams per liter, that's represented on the very top row there, and let's say the water temperature was 6 degrees Celsius, that will result in the water being 64% saturated with dissolved oxygen. Let's say you got a DO reading of 10 milligrams per liter and the water temperature was 9 degrees Celsius, that would result in an 87% saturation of dissolved oxygen. The introduction of oxygen into the water can exceed what's being taken up by organisms or released into the atmosphere, as represented on this data sheet that we give you. Sometimes if you get higher dissolved oxygen reading and the water temperatures are cooler, the amount of oxygen that's being taken up by the organisms isn't quite enough to take up what is in the water, and therefore the water can be what's called supersaturated with dissolved oxygen. We have healthy DO percent saturation levels for different streams with respect to where they're located in the state. Because in the Ozarks, generally streams have a high gradient, therefore they're generally flowing faster, and they have a rockier bottom. That means that more aeration is happening in these streams, and so the DO percent saturation for those streams is considered healthy if it's 80% or greater. Whereas in prairie streams or in northern Missouri, generally these are low gradient or slow moving streams. A DO percent saturation of 60% or greater is considered healthy for those streams. Alright, so the next parameter we're going to discuss is pH. pH stands for potential hydrogen or part hydrogen ions. Water contains both hydrogen and hydroxide ions. pH measures the hydrogen concentration on a scale from 0 to 14, and water that contains equal parts of hydrogen and hydroxide ions is considered neutral, which is represented on the scale as 7. Water that contains more hydrogen ions than hydroxide ions is acidic and has a pH of less than 7, while water that contains more hydroxide ions than hydrogen ions is considered more basic and has a pH that's greater than 7. So the pH scale is interesting because 1 unit of change on the pH scale represents a 10-fold change in hydrogen ion concentration. For example, if you have a pH of 7 reading and then you go to a pH of 8 the next time you go out and sample, that means that the water has become 10 times more basic than it was the last time you were out there. And if you went from a 7 pH to a 9 pH, that means that the water is 100 times more basic. This is also why rounding and averaging different pHs doesn't work because of the logarithmic scale. Depicted on the right is a scale of pH of water as it goes up and down. On the top part of the scale, whenever water is becoming more basic, it can become lethal to humans and aquatic life. And at the 9 to 10 range, ammonia, a nitrogen-based nutrient, starts converting to a toxic form for aquatic life. And then at the bottom of the scale, whenever the pH becomes more acidic, it can become lethal to aquatic life as well as humans. And it also allows for metals like aluminum and copper to become bioavailable within the water and taken into organisms. Normal stream water ranges from a pH of about 7 to 8. And in Missouri, our water quality standard for pH has a range of 6.5 to 9. And this is due to the underlying geology that we have in much of Missouri, that is the karst geology. It acts as a buffer for pH. Generally, pH outside of this range is going to become toxic to most aquatic life. Now let's talk about dissolved solids. Conductivity is the measure of the potential electrical current that can pass through water. It's a general indicator of dissolved solids in a stream where more dissolved solids is going to equal a higher electrical potential conductivity. We measure conductivity in microsiemens per centimeter, represented by the mu symbol, capital S/centimeters. Here are some common substances that make up dissolved solids in a stream. Since conductivity is a general indicator of water quality, any substance that can add to the electrical conductance in the water could be represented in a conductivity reading. Therefore, conductivity is often used as a screening tool to determine how much solids are dissolved in the water, not exactly what specific dissolved substances are in the water. So small amounts of certain kinds of dissolved solids, such as some metals, can cause significant changes in the conductivity itself. Currently, Missouri has no water quality standard for conductivity. Different streams have a variety of natural background conductivity. For example, Pickle Creek has an underlying geology of igneous rhyolite and granite. This type of rock is less dissolvable than limestone, which makes up our karst environments. The Big Piney, its underlying geology is dolomite and karst. This dissolves more readily, therefore it has a higher background reading of conductivity. In general, the larger the water body, the increase in the surface area of the watershed, therefore an increase in point and non-point source impacts that could potentially be influencing the conductivity of the stream. For example, Missouri River is a very large watershed. A lot of point sources and non-point sources are coming into this stream. Therefore, the conductivity readings and ranges of that are going to be higher simply because of the size of the watershed. Chloride is a substance that can affect your conductivity readings. Chlorides are salts that result from the combination of gas chlorine as well as various metals. Some have heavy metal additives, so other harmful substances might be present within chloride. combinations as well. Some sources of chloride can be road salt, especially when applied in the wintertime. And you can imagine that as vehicles travel at higher speeds, the distance that these salts can travel can increase as well. It can also come from fertilizers, underground aquifers, water softeners, storm sewers, animal feedlots, and wastewater treatment discharges. Here's a graph showing chloride spikes on a specific creek in St. Louis called Mailing Creek. In general, chloride spikes can occur in the summer whenever evaporation of the stream is greater than the precipitation that's coming in. It can also occur in the spring and fall with fertilizer application, and it can also occur in the winter with snow and ice melt. High levels of chloride can become toxic to aquatic life because it interferes with the osmoregulation in these freshwater organisms. There are, though, some invasive species that are more tolerant and therefore can outcompete native aquatic species. In Missouri, we have water quality standards for chlorides that are acute and chronic. The chronic level is 230 milligrams per liter and the acute is 860. We also have a drinking water standard that's 250 milligrams per liter. Because chloride can become an issue for aquatic life and drinking water sources, cities have developed some best management practices to help mitigate these issues. There are rate adjustments on salt spreaders that will help cities save money from not over-applying salt and decrease the amount of chlorides potentially running off into surrounding streams. Brine pretreatment is when a salt solution is spread on the surface of roads rather than salt crystals. There was a project conducted in the St. Louis area in the winters of 2016 to 2017 and 2017 to 2018 that outlines and describes how this method of road salt application could be a more effective way to reduce chloride runoff. In general, chloride isn't a problem for all streams, and we require volunteers to be a level 2 monitor in order to measure for chloride. Now we're going to talk about nutrients and how they affect stream systems. Nutrients like nitrogen and phosphorus are generally good for a stream whenever they're in balance, but whenever nutrient-rich runoff enters streams and lakes in excess and more plants and algae grow in the stream than what is usually there, this can cause what are called algal blooms, and they decrease the amount of oxygen by them being there as well as by their decomposition than what's available in the stream or the lake. So with this lack of dissolved oxygen, fish and other aquatic life might suffer and die. Nutrients can come from natural sources as well as sources from humans. Natural sources are things like just leaves falling into the stream, organic decay, and different rocks and soil that have phosphorus inside them. And then human sources can be runoff from animal production or fertilized fields and lawns. They can also come from storm drains, poorly functioning septic systems, wastewater treatment plants, or combined sewer overflows or CSOs. Combined sewer overflows are whenever the sewer system and the storm drain system are connected, and so whenever it rains a lot, those two overflow together, and the excess nutrients that comes from the sewer side of the combined sewer overflow can provide excess nutrients running into the streams as the water flows downstream. Nitrogen is an element that's essential by all living organisms, and all organic matter contains nitrogen in some form. And there's different kinds of bacteria that can break down organic matter. Nitrogen, its most stable form is nitrate. So it goes from ammonia to nitrite to nitrate. VWQM monitors measure nitrogen in the form of nitrate, which is expressed as NO3-N, or nitrate as nitrogen. Since nitrate is the most stable nitrogen compound, it can be an indicator of excess nitrogen loading. So here's some average nitrate values in a few different streams within Missouri. So starting with the Mississippi, you can see that its average value is a lot higher than all the other ones. And that's because the Mississippi is drains a super large watershed. It covers 43% of the entire continental US, and that consists of over 1,245,000 square miles, including all parts of 31 states and 2 Canadian provinces. So you can imagine there's lots of sources of nitrogen that drain into the Mississippi River. The Chariton drains approximately 800 square miles. This river is channelized, meaning that it's been straightened, and the stream drains a primarily agricultural area, so it's got a pretty wide range there. The Pomme de Terre drains an area that's comprised mostly of pasture hayfields and woods. So you've got a lot of potentially nutrient from those pasture hayfields and the wood, so it's not quite as big of a range as the Chariton. And the Jack's Fork is a relatively small impacts from point sources in the watershed, and it has an excellent vegetative cover and riparian zone. So because it has a really good riparian zone, this range is almost next to nothing. So it's stopping a lot of the nutrients that could potentially be coming in, even though the average value is higher than the Pomme de Terre. So that's just a good summary and understanding of nitrate and different streams in Missouri. For Missouri in general, we do not have water quality criteria for streams except for streams that are a drinking water source, and that is 10 milligrams per liter. Another form of nitrogen that you can find in streams is ammonia. It's the only nutrient that's directly toxic to aquatic life. Its toxicity is dependent on the pH and the temperature of the stream water. So as the pH increases, ammonia is going to convert into a toxic form. This generally isn't a problem for all streams, and we require that volunteers become level 2 trained before they monitor for ammonia. Phosphorus is a nutrient that occurs naturally in streams and lakes through the weathering of rock and the breakdown of organic matter. It's a limiting nutrient for plant growth in a body of water in Missouri, so therefore very small additions of this element can cause pretty large algal blooms. In instances where phosphate is the nutrient that limits plant growth, point source and non-point source pollution high in phosphate may stimulate the growth of aquatic plants. It's not a problem for all streams, and we require volunteers to be a level 2 before they monitor for phosphate. Alright, let's talk about water transparency. Transparency is a measure of how clear the water is. Suspended matter in plankton can cause a cloudy or murky or green color to the water itself. And so we give volunteers what are called these transparency tubes shown on the right. You fill it up with water and then look down the tube. You let out the water and then stop it as soon as you can see the bottom white and black disc that's at the bottom of the transparency tube. We measure the depth that the light can penetrate the tube in centimeters. The larger the depth of the reading, the higher the transparency. So if you filled up this tube and as soon as you looked down the tube you could see the bottom, then you would measure all the way up to the top of the tube, and that would be your transparency depth. So again, the larger the depth, the higher the transparency. So why exactly do we care about transparency? Well, let's say a body of water is very turbid or has very low transparency. That means that all the suspended sediment is going to block the light that's needed for vegetation that could potentially be growing in the stream. These suspended solids also absorb heat and increase the water temperature. That's going to affect your dissolved oxygen. Some sediment, as it settles, can also bury fish eggs as well as benthic macroinvertebrates. So these are all ways that transparency matters in the water column. When there is erosion in a stream system, it decreases the transparency because of the increased suspended solids that are in the water column. So here's just a picture of a stream that may be impacted by erosion. The water become clear and then turn murky as all of the soil that was along the banks from upstream areas are now suspended in the water column. There can be areas where monitoring for transparency can be really valuable. Areas that are being developed currently, so if there's a lot of construction in a certain area where a lot of sediment would be stirred up and run off, areas that might not be adopting best management practices to prevent soil erosion, and maybe downstream from different quarries or gravel mining operations. Here's a water chemistry reference table that goes over all the parameters that we've just talked about. It gives you sort of what it is, some natural readings that you'll see, some readings that might be cautionary or problematic, some possible sources or influences of that parameter, and maybe what you can or can't do about it. So this is a really good resource that you can find on our website and print it off and take it with you whenever you go out and monitor. Alright, we're going to talk about the data sheet for collecting water chemistry as well as briefly go over some methods. This is the water chemistry data sheet. It has all the same header information as all of our other data sheets. You'll put your site number, stream name, and all the other location information as well as your name, stream team number, and anyone who's gone out with you. You'll also have a place to put weather conditions, and this can be generally just cloud cover, and then you'll have a spot to put your nitrate result as well as any expiration dates of reagents that you use. You'll put temperatures like air temperature and water temperature, and then you'll have a dissolved oxygen reading as well as corresponding solution expiration dates. You'll have a pH result, conductivity, and transparency. The bottom half of the data sheet is more for level 2 monitors, so these are parameters like the chlorides, hardness, and phosphate and ammonia. You'll also have a section for comments, just like all the other data sheets, and if you've seen fish present at your site. The back of the data sheet, just like all of our other data sheets, are very useful if you're confused about how to monitor things or if you have just general questions about what different parameters are and what they mean. So I'd highly recommend reviewing the back of the data sheet if you haven't monitored in a while or if it's your first time going out. Alright, I know I've already said reagent already, but reagent is just a substance that's used in a chemical analysis. And another definition that you might want to know is what's called a meniscus. A meniscus is the curve in the upper surface of a liquid. Whenever we read a measurement from a small thin cylinder, it's going to have this meniscus. And you're going to want to read the measurement from the bottom of that meniscus within the container. Whenever you're monitoring for temperature, you're going to want to place it in the shade and allow it to acclimate for several minutes. And then you're going to want to take that air temperature while it's still in the shade. And the same goes with water. You're going to want to place the temperature gauge in the stream, in the shade, and allow it to acclimate for several minutes. And then you're going to want to read the thermometer while it's still in the water. This is what our nitrate test kit looks like and all the different materials that come with it. So you'll have a sample bottle where you'll take a sample of water from the stream and you'll fill it into this test tube, and then you'll add these two different reagents that have expiration dates into the test tube at different times and shake it up at different times, and then you'll eventually put a sleeve over the test tube and wait for it to finish the reaction, and then you'll compare it to these color changes that come in this color comparator case right here. Some things to remember about the nitrate test kit is that, like I previously mentioned, it's a color comparator that gives a range of nitrate depending on how dark the color is. It's time sensitive, so it's not something that you're just going to want to leave sit and then go do something else necessarily. You're going to want to make sure that you set timers and are on top of the procedures and are paying attention to them. The procedure is also light sensitive. That's why it comes with a sleeve. So if it's exposed to outside light, it could inhibit and color the nitrate result differently than what it should. And also to note that the normal range for nitrate is less than 1 to 2. So if you're consistently seeing above that, then I would encourage you to reach out to us so that we can maybe see if there's some excess nutrients input coming into your stream. This is what our pocket pH meter looks like. It's fairly small and has a glass bulb on the end of it that will measure the pH for you whenever you put it in the stream. Some things to note about the pH meter: sometimes there can be a precipitate that sort of crystallizes and grows at the end of the probe. It's pretty harmless. You can just soak it in water for a few minutes and then the crystal should just dissolve in the water and you can dump it down the drain. You don't want to submerge the probe above the cap line, and you're going to want to calibrate it within 12 hours of sampling. And then whenever you put it in the stream, you're going to want to let the meter stabilize while it's in the stream and then record that value. Again, the normal range for pH is 6.5 to 9 in Missouri streams. Our conductivity meter looks almost exactly the same. Instead of a glass bulb, it has 2 probes at the end of it that measure the microsiemens per centimeter. Some things to note about the conductivity meter is that if it's having trouble calibrating, you can soak it in water with a drop of dish soap for a while and then rinse it off, and hopefully that will help it calibrate. If it doesn't, then please contact us. We'll help you get a new conductivity meter. A lot of times water needs to be flowing or moving through these probes in order for it to work properly. Reminder to calibrate within 12 hours of your sampling event, and if it ever displays a group of solid dashed lines, then that means that the sample is over range, and you can just write over range on the data sheet if that ever happens. And again, just like the pH probe, you're going to want to wait until the reading stabilizes while it's in the stream and record it there. This is what our dissolved oxygen kit looks like and all that it comes with. So it's going to come with 3 different reagents here. They're called DO1, DO2, and DO3. And then this is the sample bottle that you collect the stream water with, and you'll add these packets to it and eventually transfer it into the smaller glass bottle using the test tube, and then you'll perform a titration to change the color of the water from a slightly orangish yellow to a clear color. For the dissolved oxygen kit, again, the final reading is a titration, so you're going to add a solution drop by drop to create a color change, and the amount of drops that you add is the amount of dissolved oxygen in milligrams per liter of that sample. The unused DO packets can go in the trash, otherwise the waste can be collected by the stream or discarded on the banks. And the normal range for dissolved oxygen, again, is between 5 and 15 milligrams per liter. Some things that you're going to want to do before you go out and monitor is to check and make sure that the reagents and the solutions that you have aren't going to be expired by the time you go out and monitor. You don't want to do this well in advance because you'll have to reorder them online from our Stream Team website. Something else is you'll be submitting the data online, so you're going to want to double check your data sheet and make sure that your data looks good before you enter it. And if you are wanting to pick up another site by either adopting a new site or creating a new site, you can email streamteam@dnr.mo.gov with a map and a site description. After you use the dissolved oxygen and nitrate test kits in the field, you're going to have waste from them. This waste can be stored or containerized and then poured down the sink after you are home. And then after a while, some of your liquid reagents as well as powdered reagents are going to become expired from the DIEL kits, nitrate kits, pH, and conductivity solutions. All those can be either thrown in the trash if they're packets, or they can be poured down the drain while you can flush with ample cold water. This is just a reminder that we don't want pets or children getting near these chemicals, as they are not good for consumption or getting, in the eyes and mouth area. Some ways you can get involved and next steps you can take is to register for a field training. Go to mostreamteam.org to see our announcements where you can sign up for and register for field trainings in your area. And then we will be shipping equipment out to you after you attend the level 1 field training, and you can start monitoring. So here's how you can contact us. Again, our website, we have 2 different emails, streamteam@dnr.mo.gov or streamteam@mdc.mo.gov. We're on YouTube and Facebook, and you can call that number as well to get a hold of us. Thank you.